All right, let's dive right in. We're going to talk about some of the most cutting edge medicines out there today. But behind these incredible new drugs, there's this hidden manufacturing challenge. I mean, we're looking at therapies that could totally revolutionize how we treat diseases, but there's a catch. Scientists first have to solve a really tricky puzzle. So here's the central question, right? How do you actually manufacture a medicine that's designed from the ground up to be complex? It's this amazing clash where brilliant new ideas run smack into the hard realities of making this stuff. And, it's a really fascinating problem to tackle. Okay, so to really get a grip on this puzzle, we've got to meet the hero of our story. It's this groundbreaking tool in medicine called the bispecific antibody. And trust me, it is a total game changer. So why is it so special? Well, to put it simply, it's a drug that can do two completely different jobs at the exact same time. Just imagine a single molecule. With one arm, it grabs a cancer cell, and with the other, it grabs one of your immune cells. It literally pulls them together so the immune cell can do its job. That's the kind of power we're talking about here. Incredible, right? So the official term is a bispecific antibody, or BSAB for short. And it's really an engineered marvel. See, its structure is what's so special. It lets it bind to two different things at once. Now, you have to understand, this is a huge leap forward. Traditional antibodies, they can only grab one thing. This dual action ability, well, it just opens up a whole new playbook for treating diseases. But, and there's always a but, right? Here's the twist. The exact same complexity that makes these things so powerful, well, that's also what makes them a nightmare to produce cleanly. Trying to get a final product that's pure, safe, and effective, that's a massive challenge. It really is a purification puzzle. So when they're being made, a whole lot can go wrong. You end up with these, well, let's call them the villains of the story. You've got mispaired species. That's when the different antibody arms just don't snap together the right way. You get things called half antibodies, and you get aggregates, which are pretty much just useless clumps of protein. And every single one of these impurities has to be filtered out. And this really shows you just how big the problem is. If you look at the older, simpler antibodies, monoclonals, the level of these junk variants is pretty low. But with bispecific antibodies, the manufacturing process creates way higher levels of all this unwanted stuff. This difference, this purity gap, is the absolute core of the challenge that scientists are trying to solve. So the big question is, how do they do it? How do they clean this up? Well, it turns out the tried and true methods just aren't cutting it anymore. For these next-gen drugs, scientists have to go way beyond the old industry gold standard. You see, for decades, the go-to tool for purifying antibodies has been this thing called protein A. It's been the absolute workhorse of the industry. Think of it like a super specific magnet that grabs onto antibodies and separates them from all the other junk in the mixture. Super reliable. But what happens when that trusty standard tool just doesn't work as well? That's the problem. Some of these new bispecifics are designed in a way that actually changes the part of the antibody that protein A is supposed to grab. So that magnet isn't as strong. It's like the net suddenly has bigger holes and the things you want to catch are slipping through. It forces researchers to get really creative and come up with entirely new solutions. And this is where things get really cool, because this challenge has led to some much smarter, cleaner strategies. Instead of just relying on that one old tool, scientists are now using this multi-step, really data-driven approach to hunt down and get rid of every last bit of impurity. All right, so this modern path to purity, you can pretty much break it down into 3 key steps. First up, they're using new affinity tools. Think of them as newer, better magnets that are way more selective than the old protein A. Step 2, they get a handle on those protein clumps, the aggregates, much earlier in the process. And finally, there's a precision polishing step at the end, which is designed to remove any really stubborn impurities that are left hanging around. And this polishing step, this is where things get really sophisticated. Scientists have a whole toolkit of different methods, mostly different types of chromatography, which is basically just a super high-tech way to filter molecules. For example, they might use something called ion exchange, which is amazing for telling apart variants that are almost identical. Or they could use hydrophobic interaction for other specific leftovers. And then there's mixed mode, which is kind of like a Swiss Army knife for tackling really complicated mixtures. It's all about picking exactly the right tool for the right problem. So, okay, we've gone through the problem, and we've looked at these really clever solutions, but you might be wondering, why does all this technical detail actually matter? Well, it all boils down to one simple, crucial thing. The path to purity is ultimately the path to the patient. And really, this is the bottom line. This isn't just some interesting science problem in a lab. For any drug to even get into clinical trials, it has to meet incredibly strict standards for purity and safety set by regulators. Solving this purification puzzle isn't optional. It's an absolute must if these life-changing medicines are ever going to get approved. Which kind of leaves us with one last, really fascinating thought. As scientists get smarter and better at solving these incredibly complex purification puzzles, it just makes you wonder, right? What new cures, what new breakthroughs for things like cancer or autoimmune diseases? What's waiting for us just on the other side of these challenges? The potential here is just absolutely immense.